this, initially with RNA-seq. Now we've done low-pass whole genome on most of the collection, and also exome, both from somatic cells and from the leukemic tissues. And so what this shows here, and what we mean by group comparison, is that we've really gone quite rapidly in verifying what are the outliers in terms of gene expression in this disease, and characterizing it through each subgroup. And I'll give you an example of this today for the RUNX AML. Basically, that gives you a beautiful signature of the disease. And then when you get to so many specimens, you can start to try to predict new subgroups, which I will not be discussing today, but as you will see in the coming months. There's also a lot of work that's been put through the program on being able to grow the cells ex vivo, and we continue to put a lot of efforts to find ex vivo conditions that allow us to maintain the precious leukemia stem cells and prevent their differentiation, such that when we use compounds on these cells, we really are in a fairly good level of comfort that we're looking at the kind of response that the cells would have in vivo. The third pillar is what we call pattern recognition, and we've realized by putting lots of compounds on a lot of primary specimens that there's a very strong tendency for many of these compounds to give us the same pattern of sensitivity. So in graphs like these that you will see, each line represents a compound, a different compound, and each of the vertical point from 1 to 20 in this case represents a specimen, a patient specimen. And as you can see, for example, in this first CCC that we call for compound correlation cluster, we find that there's a lot of compounds that give us the same pattern. These specimens here being the sensitive one, the one here being the resistant one. And what was very interesting, which rapidly emerged from these studies, is that sometimes you have a mirror image like this one. For example, specimens that are sensitive to this compound are more resistant to this one, and vice versa. And so this gives us the possibility to start to test two compounds simultaneously, what we call synthetic fingerprinting. And although I will not be discussing too much about this today, this really works well, and that will be for another presentation. And the last pillar is really the ability to integrate all of the genetics, the chemical interrogation, and the clinical data into a suite that we initially call Mystique, where we basically can query at any time the gene response to a compound, or the clinical data around mutations, or around the combination of a mutation and chemical response. Okay, so let's focus today on the RUNX1 subgroup, and this will be a bit of a Laura Simon presentation. I know Laura is probably online, so I just want to say that a lot of what's going to be shown today was basically done through the PhD thesis by Laura, and want to congratulate her for her beautiful work. So you're looking here at what we call an icicle representation developed for Mystique, and this is basically showing that each peak here, each of these peaks are a series of compounds that correlate together. And when you basically touch this line here, the correlation is 100%, 9.75% here, 97.5% here. And in fact, today we're going to focus on this glucocorticoid peak here. Okay, so the first piece of work, which I'll quickly go through because it's basically just to show you what Laura just finalized recently and published in Blood, which should come out shortly. It's basically this high frequency of germline RUNX mutation in the RUNX1 AML patients in the Lucégène cohort. So this was the first Lercégène cohort, not the 700, but the 430 patients. And Laura initially identified 67 of these that are RUNX mutated, 44 were validated. And then by applying specific filters for the presence of mutation in tumor DNA and the absence of mutation in buccal DNA, she identified 16 patients that have mutated RUNX in buccal DNA. And then through an additional filter was able to filter out 4 of them to really make sure that when she was calling a germline mutation, it was really a germline mutation. And this was done mostly by confirming the somatic status of at least one additional leukemia-related mutation in the germline versus leukemia specimen. And she really needed that criteria for this to be called germline. And this is just an example of this. I won't spend time today on how this was done for distinguish somatic versus germline. I just showed you the filters that were used. And basically, to make a long story short, what she identified is really 12 of these patients that had, in fact, germline mutations. And this was quite of a surprise for us. And as you can see, when you look at some of the characteristics, the germline patients had a non-significant tendency to be a bit younger, higher white blood cell counts, but the rest was basically pretty much the same, both in terms of genetic groups, FAB, etc. Now, when you look at age of AML onset versus the type of mutation, the germline being in red in this bar graph, and the somatic ones being in black, you immediately see that there was a strong tendency for those that are younger than 50 years old to be germline, whereas those that are older to be somatic. There was a strange occurrence in this series of this lady here and her young son here, in fact, to have a germline RUNX1 mutation along with a second mutation on another gene. And in fact, what's very interesting is that he developed AML at a much younger age than his mom in this case, whom we treated first, and he was treated second in our center, in fact. Okay, here you're looking at the most frequent mutation in germline versus somatic RUNX. I mean, it's a small series. It's tough to make any intelligent conclusion. Maybe a tendency to have more NRAS mutation in the germline than the somatic, but that's all we could say with this. And so the conclusion is basically what I just said. Okay, let's now focus on this glucocorticoid CCC. So Laura published a couple of years ago the chemogenomic landscape of RUNX1 mutated AML, and I just want to highlight some of the few findings on how she came to identify that glucocorticoids were quite effective in a subset of these RUNX1 AML. So basically, she got this initially from 20 different primary specimen patients that we treated with 5,000 compounds in a screen. And in fact, looking at 3 different glucocorticoids here of many, you can see that we found that the most sensitive patients, those in fact shown by the red dots here, were enriched and strongly enriched for RUNX1. And so we tested, Laura tested many more patients that are either RUNX mutated or RUNX wild type or 821s, and basically identified that a significant proportion of RUNX1 mutated patients were indeed responsive to glucocorticoids. And what was very interesting is that they were extremely sensitive, like low 2-digit nanomolar range. And the other patients were usually very resistant, even 10 micromolar with no response. So it was really an all or nothing phenomenon. And then she dissected the mutational profile, nothing very different from what is known here. But something quite interesting, which I mentioned earlier, is that we saw a very distinct signature for these RUNX1 mutated patients. And then when you start to look at genes that are correlated, well, they're all correlated as part of the signature. And in fact, the strength of the RUNX mutated alleles, for example, if you have NS, FS, a nonsense and frame shift, you have higher levels of glucocorticoid receptor, whereas if you just have a mis or a missense allele heterozygous, you have a tendency to express lower levels of the glucocorticoid receptor called NR3C1. So NR3C1 is obviously part of the signature that Laura identified. So another thing that was clear, these are again data from Mystique, is that if you look at the RUNX1 mutated specimens, you will see that they express quite high levels of NRC31. As you see in the low expressors, you have a tendency to deplete for RUNX1 AML patients. Now, Laura took almost 250 primary AML specimens and tested them with true glucocorticoids, now dexamethasone, flumethasone, and hydrocortisone, and really did EC50 on these. And again, looking at a lot of non-RUNX, including RUNX specimens, 33 versus 220 essentially. The mutations that were mostly enriched were RUNX1 for the 3 different glucocorticoids, but also SRSF2 and CBP alpha, which are frequently co-occurring mutations with RUNX1. So she also found that the allele dosage dictates the glucocorticoid response. And again, it has a strong tendency, this is a log scale, it has a strong tendency to be in all or nothing responses. And she could identify some mutations that are associated with glucocorticoid sensitivity and other mutations that were associated with glucocorticoid resistance. Now, what she also found is that if she generates shRNA and reduces the levels of RUNX1 with 3 different SH to about 50%, this is a Western blot here, you will see that cell lines that are normally resistant, like AML5, AML1, AML4, and these 2 guys suddenly become sensitive to these 3 glucocorticoids, to dexamethasone in this case. Whereas other cell lines, like monoMAC1, remain refractory to glucocorticoid, even if you downregulate levels of RUNX1. So there's a level of complexity highlighted both by these knockdown experiments, but also by the primary specimens that Laura studied and that I showed you before. And there is a certain tendency with the strength of the allele giving you sensitivity, but there's something else as shown by these SH experiments here. Now, there's another interesting fact, which I guess you could guess from what was shown before, is that if you knock down RUNX1, you have a tendency to increase the expression level of the glucocorticoid receptor NR3C1 as shown here. Now, what's very interesting is that when you knock down RUNX1 in a resistant cell line, in this case, it becomes sensitive to dexamethasone. These are AML5 cells, but there's absolutely no difference on their sensitivity to other chemotherapeutic agents such as cytarabine and 6-thioguanine. So this is specific to dexamethasone. Okay, I've only put this slide for reference. Now, another thing that's very important is that if you knock down the glucocorticoid receptor on which dexamethasone binds, as shown by these 3 SH here, as shown by the Western here, and you grow these cells in the presence of dexamethasone, now they don't care anymore. So the impact is really occurring through the glucocorticoid receptor. And in fact, if you initiate the culture with a 50-50 transduced and untransduced cells, and you grow them after 7 days, it's only the GFP positive transduced cells that have survived dexamethasone treatment. So we're really on target in this effect here on the glucocorticoid receptor. And last but not least, the glucocorticoid receptor can be immunoprecipitated with RUNX1. And there's more of this in the presence of dexamethasone than in the absence, and it could be because there's more of the receptor. Okay, now I'm going to spend the rest of the time on this more recent point, which is a paper in preparation, and it's really trying to dissect the sensitivity to dexamethasone. Now, our logic here is that glucocorticoids, they hold a good therapeutic potential, certainly against ALL. I would say they probably remain the cornerstone of treatment in ALL. In AML, as you know now, there's mostly only a subgroup of RUNX1 mutated specimens that are sensitive to glucocorticoids. So we postulated that if we could understand this better, maybe we would be able to use glucocorticoid more extensively in the treatment of AML, a drug that we all know how to use in the clinic. So the mechanism of action of the glucocorticoid receptor is summarized on this slide, and very importantly, what it does upon binding to glucocorticoid is that it's translocated into the nucleus, where it has important effects. First and foremost, it has a transactivation effect on glucocorticoid response element, or GRE, and it has a transrepression effect through various mechanisms. There's also non-genomic effects, and there's also mitochondrial effect, but for today, we'll focus on this. Now, one of the first things we did is basically looking at glucocorticoid-induced transcriptomes in resistant and sensitive cell lines, and also engineering a sensitive cell line to become resistant. The second approach was really to do a CRISPR-Cas9 chemogenomic screen using 2 resistant cell lines in the presence of dexamethasone. And finally, doing a chemical screen to identify synthetic lethals to dexamethasone. So let's go through some of this data. So in the first approach, we find that glucocorticoid-induced transcriptional response can be observed both in sensitive and in resistant cells. So we've used the resistant AML5 cell, we've used the sensitive AML3 cell, and we've used the engineered resistant AML3 cell in which glucocorticoid receptor was knocked down by SH. So shown here. We treated the cells as indicated here with 100 nanomolar of dex, and then harvested them and did RNA-seq. And one of the first things that you can see both in the resistant and sensitive cell is that there's quite a few genes that are upregulated, fold change is shown here. Now, the sensitive cell seems to show, and this is time, seems to show a much larger number of genes that are upregulated. Genes that are downregulated are of about the same number. Now, what is very nice, and it's one of the cleanest experiments I've seen basically, is that if you knock down the glucocorticoid receptor, you see almost no genes that are either up or downregulated. So if you look more into these genes by a weighted gene correlation network analysis, and you see what you have, you see a lot of neutrophil activation. And if you look more closely into the sensitive AML3 versus the resistant AML5, these are the various time points of DEX exposure from 0 to 48 hours. What we see a lot, at least in this cell line, is activated macrophage markers that go up, and also TF involved in macrophage polarization. Now, this suggests that it might be linked to, this impact might be linked to macrophage differentiation. And there's one gene in particular which is CD163, which can be used obviously in screening conditions. And you see here fold induction upon DEX treatment in these cells. And if you give an inhibitor to the GR receptor, you in fact, by fact, you see that the protein CD163 is not very well expressed. But if you look at different doses of DEX, you see that even at 10 nanomolar, there's already maximal expression level. The resistant cells do not upregulate CD163. So this is interesting, was given to us by a patient from the Princess Margaret at Toronto. This patient was a 78-year-old refractory AML, RUNX1 mutated, and by testing this in vitro with our conditions, they in fact realized that the patient's cells were sensitive to dexamethasone, and as you can see, upregulated CD163 quite significantly, and in the CD34 negative compartment, compatible with monocytic differentiation as well. Okay, now if you look at several cell lines that are GC resistant or GC sensitive, you will see that the sensitive cells do express, and it varies, some levels of CD163, whereas the resistant cells do not. This is again 10 nanomolar of dexamethasone. So the conclusion from this is that even though glucocorticoid is active, at least transcriptionally in the resistant lines, it fails to activate in these lines the transcriptional program associated with monocytic macrophage differentiation. The top-regulated genes in the sensitive lines are enriched for macrophage differentiation polarization towards the M2 phenotype, and CD163 is strongly induced by glucocorticoid in the sensitive lines, but not in the resistant ones. Okay, so the second approach I mentioned was basically doing CRISPR-Cas9 screen in collaboration with Mike Tyers here using a library developed in his laboratory. And for this, we use, Laura in fact used two resistant cell lines, the AML1 and the AML5, exposed to two different doses of dexamethasone and screen at different time points up to 14 days when the culture was essentially killed. Of course, what we found there in both cell lines is that the glucocorticoid receptor, or NR3C1, showed the strongest rescue interaction with dexamethasone. That, of course, we knew from our previous work. Now, it was very interesting, and I see that John is here, and I think he'll be interested by this. ZBTB16, or I will call it PLZF, which is a lot easier for me, was identified as our top hit in terms of lethal interaction in both of these screens. So if you look at PLZF expression in human cells, this is just our own collection, but it's been done by many, many people. So these are the 430 primary specimens. This is TPM value, RNA sequencing. So B cell, CD34, as you can see, it's highly expressed in primitive cells, goes down with granulocytic differentiation. It's expressed in some TALL and some T cells. Now, if you look at RUNX1 mutations for this, we don't see a real difference between the mutated and the non-mutated cells in terms of PLZF. So PLZF associates with repressors to block myeloid differentiation. This has been studied by many groups. It's involved in a translocation promyocytic leukemia, the famous PLZF RARA. As most of you know, it acts as a transcriptional repressor and COL3 adapter for substrate binding and ubiquitination. And it's upregulated in glucocorticoid-resistant ALL patients, and downregulation of this gene may show increased sensitization to glucocorticoid in ALL. So we looked at this more carefully. So one thing that we see is that PLZF is highly expressed in the glucocorticoid resistant AML cell line and not so in the sensitive, although when we look at a larger panel, it's not so black and white. And it's obviously expressed in the 2 cell line that we used for CRISPR-Cas9, although a little bit higher expression in the AML5 than the AML1. So if we knock down PLZF in both lines with 2 SH as shown here, we seem to sensitize these cells to DEX treatment. This is the AML5 data here, and this is the AML1 data here. Now, what is very interesting and akin to what I showed you for the glucocorticoid receptor, the knockdown of PLZF does not affect the response to other compounds. So again, as you can see, it's quite a dramatic effect when you look at 2 different glucocorticoids on the AML5. We're talking, up to 2 logs in the AML1, so it's a huge effect on IC50. But if you look at other compounds like cytarabine and 6-thioguanine, again, PLZF knockdown has no impact. So it's like the glucocorticoid receptor, it's highly specific to glucocorticoids. Now, we used this famous transactivation DCAS approach to endogenously reactivate PLZF into AML3 sensitive cells. This is the Western blot showing the SG control and the SG PLZF endogenously overexpressed. And what you can see in this case is that now, in contrast to the knockdown, the cells become significantly more resistant to 3 different glucocorticoids. And we're talking here 3 logs. Again, no impact on non-glucocorticoid compounds. So this validates the results of the CRISPR, but it also suggests that modulation of PLZF can have a tremendous impact on glucocorticoid sensitivity in AML. Now, the third approach, the question was, can we target gene sensitizers using small molecule to promote glucocorticoid? I'm not sure I like the word toxicity, but let's say maybe monocytic differentiation or maybe apoptosis. But so, as you know, PLZF is a difficult protein to target. However, epigenetic modifiers have been shown to release the repression by PLZF. And we really performed here a combinatorial screen with DEX and a 10,000 compound library that we obtained from a pharma partner in glucocorticoid resistant cell line. And lo and behold, we were able to find small molecules that synergistically enhance the impact of dexamethasone and others that will block the impact of DEX on this line. So, but that's where we stand with this project. So our second conclusion from this is that we really performed the first functional genomic characterization of GC response in AML-derived cell lines by CRISPR. As a proof of concept, the screen identified the glucocorticoid receptor and several components, which I didn't show today, of glucocorticoid receptor maturation and transcriptional co-activators as positively selected genes, so genes that rescue the impact of glucocorticoids. PLZF was identified as our top negative regulator of GC sensitivity, so this was really a case of PLZF knockdown showing synthetic lethal. And we also showed that PLZF is expressed in several glucocorticoid resistant AML cell lines, and as you've seen, that modulation affects glucocorticoid response. Of course, we found small molecules that can modulate the effect on GC. It will be extremely interesting to see if, at least in preclinical models, if these molecules have an impact, and possibly also in the clinic. As per Laura's paper 3 years ago on glucocorticoid sensitivity, there's been quite a few number of people calling me and telling me that they've tried the glucocorticoids on RUNX1 mutated refractory AMLs. I have in mind at least 3, and in all 3 cases, I've heard complete remission that was induced by myeloma type treatment of these patients by dexamethasone. I haven't heard that much follow-up from these patients though. So perspective is really to compare the epigenetic marks in GC sensitive and resistant cell lines and validate the influence of GC response, to define the mechanism by which PLZF affects GC response, and to validate the synergistic effect of our screen compounds first in PDX models. So again, this was done by Laura, who's right here, and the supporting team behind her on the Sauvageau project. And I want to again give special thanks to Laura, to Mike Tyer's group for the CRISPR, to José, the co-principal investigator of the Sauvageau project and BCLQ, and Sébastien and Geneviève, our bioinformatician, our key bioinformatician in the project. So that's where I stop, and I'd be happy to take any questions.
Great, thank you so much, Guy. Wonderful presentation, and we already have some questions that I'll start with. The very first one is from Patricia Ernst. Exciting talk, Guy. Could you explain further the relationship between physical interaction and gene expression relationships between RUNX1 and the glucocorticoid receptor?
Well, hi, Patricia. Well, first, I want to say hi to all of you guys, and I hope you're all doing well in this terrible moment. That being said, Patricia, we're investigating this right now, unfortunately for us, Laura is about to leave, and she has trained a wonderful lady who's taking over, and that's a key priority right now.
Great, thank you. The next question is from John Licht. Hi, Guy, did PLZF affect glucocorticoid receptor levels? Did it affect levels of BH3 protein levels? And did HDAC inhibitors block the PLZF effects?
You know, John, I don't remember. And I know Laura is probably listening, so if she can write the answer, I'll tell you. But I don't remember.
Okay. Laura, you can actually raise your hand and I can unmute you if you do have the answer. So just let me know. I'll keep my eye out. The next question is from a patient. And her question is, in your research, have you seen RUNX1 FPD affecting the development of multiple myeloma?
I wouldn't know. We haven't looked at that. I'm sorry.
Thank you. Okay. The next question is from Wenbin Zhao. Among the 12 AML with RUNX1 mutations who were germline, how many had family history of hematologic disease? Any difference in terms of platelet counts?
Unfortunately, we didn't. I mean, the way we built the bank almost 20 years ago now, we were stupid enough at that time not to collect family history, and we don't have the consent forms to go back into this, and that was that's something that's really sad, actually. So there's only one of which we know because, you know, by sequencing we immediately realized that they were, you know, mother and son. We wish we had done this differently, so I can't answer you about the family history, even platelet counts.
Okay, we have another question from a patient. My daughter developed AML at age 12, 2 years before I was diagnosed and treated for MDS at the age of 50. We both have germline RUNX1 mutations. Is there research ongoing into the relationship on potentially telomere length or shortening in following generations? And could that be linked?
Yeah, there's, well, to the best of my knowledge, there is a lot of research in human and in mice. There's certainly no shortening in telomeres unless you have mutations that can affect this. Um, but so in this case, I would say most likely no. Um, what we've seen in instances like yours is that sometimes there were co-occurring mutations that were transmitted um, in addition to RUNX1. So that's all I can say.
Thank you. Next question is from Isabella Iasenza. Would you say that RUNX1 is interacting with the glucocorticoid receptor and binding the GREs in the nucleus to either block or enhance transcription?
I would think so. I think the answer is probably yes to that question, but you know, that work, we haven't done it, and we're starting to explore the chromatin quite a bit more if we can have a lab eventually. Yeah.
Great. Laura has raised her hand, so I'm going to allow her to speak. Okay, you should be able to speak now, Laura.
Hi, can you hear me?
Yes, we can.
Hi, Guy. Thanks for the excellent talk. Yeah, to answer John's question, we have not observed increase of the GR receptor when we knock down PLZF, for example. And we haven't checked for BH3 proteins. But for the HDAC inhibitors, yeah, I was hopeful to see an impact with at least HDAC1 inhibitor. We see a little bit of increase in sensitivity to glucocorticoids using the resistant cell lines, but we still have to perform additional experiments to conclude that. We didn't see a great improvement in sensitization using the HDAC inhibitors.
Great, thank you so much, Laura. I'm gonna mute you again. And I'm gonna move on to the next question, and that is from Grover Bagby. Were the NRC31/RUNX1 immunoprecipitates done with nuclear, cytoplasmic, or whole cell lysates?
Hi, Grover. You know what? I'm not sure. Again, you're going to have to call Laura for this. Sorry.
Laura, I've just unmuted you if you want to answer that question. Yeah, so they were performed in both cytoplasmic and nuclear. So RUNX1 is mostly seen in the nuclear fraction. So we only see the interaction between GR and RUNX1 in the nuclear fraction.
Great, thank you.
Thank you.
The next question is from Lucio Castilla. In the RUNX1 germline AML group, is the loss of the second RUNX1 allele correlated with a higher sensitivity to glucocorticoids?
Well, first, we haven't been able to systematically test these patients because, as you can imagine, Lucio, we would need to, you know, thaw a vial and basically expire very precious material. I can certainly say that there is a tendency for more severe alleles to correlate with sensitivity to glucocorticoid, and it's mostly, although not completely, but mostly an all or nothing response. So they're either sensitive 2-digit nanomolar or not. There's a few exceptions to what I said, but I guess that's pretty much the rule.
Great, thank you, Guy. The next question is from Anupriya Agarwal. Great talk. Do you know whether the glucocorticoid receptor expression in FPD patients, what it is at the pre-leukemic stage prior to somatic mutations?
No, that's a good question. And you know what the impact of glucocorticoids could have on these patients, and even on, you know, when they acquire additional mutations, can you eliminate these clones more selectively by giving them short courses of glut? I don't have the answer to that question. It's an outstanding question.
100% agree. The next question is Jianping Li. Do you know if glucocorticoid works for AML1 ETO patients or AML1 ETO cell lines?
So Laura tested the rare cell lines that exist and also quite a few patients, and the answer is yes. But again, there are resistant and sensitive patients, and unless Laura knows something I don't, which as you've seen does happen, I'm not aware of why that is. I don't think we know for the AML-ETO what's going on in terms of sensitivity versus resistance.
Great, thank you. And then a question from Alan Kentor. Is there a connection between RUNX1 deficiency and PLZF expression? If not at the RNA level, have you looked at the protein level?
So I don't think there is, and we haven't done that. These are very good questions. It's always, you know, the issue of getting patient primary specimens. They're so precious and there's so few of them, and we want to keep them for some of the chemical screens that we're doing. So so I wish I could answer that question, Alan.
Thank you. Question from Martin Ernst. Thank you for a very nice talk. I have two questions. The first one is, the proportion of RUNX1 germline mutation seems pretty high. Were there more family members in the group of germline patients? Was the somatic mutation present in all germline RUNX1 mutated patients to verify germline status? That's the first one, and then I'll read the second one.
Okay, so for your first question, the proportion of germline mutations being high, I agree with you. We were quite surprised. There's only one instance, and that we could verify by looking at SNPs, because now we've done a whole genome of those. Looking at SNPs, we see that there's a mother and son, as I mentioned, but there's also one additional couple that was related, I think, by third or fourth generation. That's it. The other ones are absolutely unrelated to the best of our knowledge. Was the somatic mutation present in all germline RUNX mutation to verify? Well, you know, the answer has to be yes, because it was one of our criteria to verify that in the mouth swabs, we had evidence for RUNX mutation, but not the corresponding somatic mutation. And in the AML cells, you had to have both. So that's how we dealt with this issue.
The next question from Martin was, was there any difference in clinical outcome between the germline or somatic RUNX1 patients, for example, in complete remission after induction therapy, overall survival, event-free survival, et cetera? That data.
Not to my knowledge. Of course, we've expanded the leucogene collection now to close to 700 patients, and that's something we're going to look more carefully. And the fact that we've done exomes from somatic and tumor and non-tumorigenic tissue will allow us to more directly identify the germline in this group, not just of RUNX, but of the other familial AML diseases. And I'll be more able to directly address that question, Martin. Thank you for that question.
The next question is from Grover Bagby. Does GATA2 knockdown, a PLZF interactor, also sensitize glucocorticoid resistant cells to dexamethasone treatment?
Of course, you know, we asked ourselves that question, and it's another outstanding question, Grover. To my knowledge, to the best of my knowledge, again, we've only tested 2 cell lines thus far, but to the best of my knowledge, the answer is no. I believe, and you can tell me if I'm right, but this monomac1 cell line is also resistant, and I think it does have a GATA2 loss of function, but I'm pretty sure, but I'm not 100% sure. so we would need to test more cell lines to carefully address your question.
Great, thank you. Another question here from Sue-Ann Fontenard. What was the average number of acquired mutations in the patient samples? I'm assuming she's probably asking between germline and somatic RUNX1.
Laurie probably remembers better than I do. It's been 3 years that we've looked at this. I don't think there's a difference, Sue-Ann. I think it's about the same in both cases. It's in our RUNX1 paper 2017.
Great. Another question, do you happen to have a theory if trans activation or trans repression is occurring when treating with glucocorticoids?
So Isabella, that's easy to answer. So the answer is that both are occurring. As I tried to show today, trans repression, we see fewer genes and we don't see difference in numbers between the sensitive and the resistant cells. Trans activation, we see more genes that are being upregulated, including the CD163 in the sensitive lines. Now, I think where you're going with this is whether or not we think that trans activation or trans repression is essential. So with some of our medicinal chemists, Laura had done a lot of nice preliminary work trying to look at glucocorticoids that have, you know, more or less trans activation versus trans repression activity. And it looks to me, and Laura could correct me, that we probably need both. But some of the molecules that were presumed to be clean in lacking either trans activation or trans repression may not be so clean when we search the literature, and that is a key program that we want to continue to develop, and which will be the subject of a grant in the coming year for sure, because you know, we know that some of the side effects associated with glucocorticoids are more linked towards one of the two mechanisms, and we would like to shy away from side effects as much as possible, as many people have tried in the past. Hope that answers your question.
Thank you. Next question is from Wenbin Zhao. Among the upregulated genes in RUNX1 mutant AML, TCF4 and RUNX2 are both among the top genes, and they are important for plasmacytoid dendritic cells. Can you comment on that?
Yeah, I agree. We saw this, and I must admit that for another reason, we find these PDC cells quite interesting in terms of enhancing NK cell activity in vivo. So I presume that you know, there's something about a repression of PDC program by RUNX1. That's all I can say, but that's something we had noticed indeed.
Great, and then we had one more question from Sue-Ann, which you alluded to already the answer to earlier, but I'll ask it again. What was the diversity of acquired somatic mutations in the germline cohort? In the two families, are there similar acquired mutations present?
You know, it's been a while, although the paper is still in press. It's been a while since we've, you know, submitted it, and I would like Laura to answer this one. I can't remember completely. I think it was quite similar, but in the two families, there were particularities. One was with CBP alpha, if I recall, and the other one with GATA2. But Laura, if you could answer this.
I've unmuted you, Laura.
Sure, yeah, thanks, Catherine. Yeah, in terms of the two cohorts, the germline and the somatic, they had similar numbers of acquired mutations. I think we had two examples, patients with P53 mutations that had lots of mutations acquired, but in general, they didn't acquire a lot of events. I think that they have most like 3 to 5 mutations each.
And Laura, what about the two families, especially the one family, mother and son, and the other guys?
So the mother and son, they both had the two RUNX1 and CBP alpha germline mutations. The mother acquired a GATA2 and the son acquired a chromosomal abnormality. Only had these three abnormalities that we could pick up with our screens.
Okay, thank you.
Thank you, Laura. All right, I don't see any other questions. We have 3 minutes left, so maybe I'll give a few more seconds in case anyone has anything else to ask. But in the meantime, I just want to express my sincere gratitude on behalf of the RUNX1 Research Program and the community at large for your time today, Guy, and for taking, for really pursuing this question around, you know, what is driving the sensitivity of these RUNX1 mutated AML cells to glucocorticoids. And it sounds like we're on a path to potentially, that you're potentially going to pursue maybe a clinical trial in the future for RUNX1 mutated AMLs, I hope. Certainly would be great to see that impact in the clinic.
Well, thank you, Catherine. Thank you all for your time, for your questions. I hope that you all stay safe, and I would like to comment that I'm also very keen about, you know, the chemical screen that was done to see if we could indeed eventually bring a series of compounds to enhance sensitivity to dexamethasone in this disease. Yeah, that would be cool eventually. If there's any other question from any of you, please don't hesitate. Just send me emails, and it'd be a pleasure to answer them.
Great. Thank you so much. Take care, everyone. Be well. Stay safe.